Genetic engineering. We're going to focus on genetic modification and cloning and GMOs. Now the two pictures there, they are photoshopped, so just a little heads up about that. Now here are 10 topics or terms that I would definitely include in index cards to help you study. We're going to be going over each one of these. You'll find them throughout the notes. Now be aware that these are not 10 terms. Some of them are terms, definitions and terms, but if you look at number 9, that includes 5 parts or 5 steps. And number 10 will include various multiple responses or answers. Let's look at the first one. Genetic engineering is the deliberate modification or change of the character traits an organism has by using special techniques to control and to manipulate to change the genetic makeup, to change the genes. So basically we're changing the DNA in a living organism to create something new. This is usually done on plants, even though it's also done on animals. The word GMO means genetically modified organism. Many times when we look at different food products or hear commercials that the food product is GMO free, meaning that it is not genetically modified. GMOs are also called transgenic organisms because you're transferring different genes from one to another. So GMO, genetically modified organism, primarily talking about plants or some living organism. And another name for that would be transgenic. An example of a GMO would be bacteria that's modified to make or produce human insulin. A long time ago, they used to take the insulin from pigs and give that to people, humans, but now they can actually inject that and have the bacteria, change the bacteria's nature, its genes to produce human insulin to give to people later without killing, I guess, the pigs. Some other examples of GMOs. The grapes that taste like cotton candy. Those are genetically modified organisms. They even were able to get fish to glow in the dark. And I think they even tried that on tomatoes to glow in the dark. Okay, here are some terms that are going to tie in with what we're going to be focusing on in genetics, but also in our next unit, artificial selection. We're going to divide this up into 3 parts: selective breeding, hybridization, and inbreeding. Then we're going to focus on cloning and how that's done. Artificial selection focuses on the intent reproduction of individuals in a population that have desirable traits. And that's the key here. Artificial selection is choosing the desirable traits that you want. So you are choosing what traits you want to pass on. So an example of that is selective breeding. So you're not really using any lab equipment here in a major sense. All you're really doing is saying, oh, this animal has a desired trait. I'm going to mate it with another animal that has a desired trait to get what I want. But this dog, this animal, this horse has a trait I don't want. So I'm not going to mate the animal that I have with the animal that has the undesirable trait. I'm only mating animals that have desirable traits, choosing the best male, the best female to reproduce those desirable traits. Examples would be racehorses, show dogs, cows with tender meat, juicier oranges. I'm choosing the best to mate to get the best. And that's the key word, I'm choosing. Okay, hybridization occurs when individuals with unlike characteristics are crossed to produce the best of both organisms. So there are 2 animals or 2 plants that are different, and then I'm mating them together. Remember, hybrid means different. Hybrid was referring to many times you're heterozygous. An example of this was Luther Burbank. They created a disease-resistant potato. So these potatoes survive the mold and were able to survive certain diseases from breaking down and destroying the plant. So we crossed a disease-resistant plant, one that survived this disease, and placed it in, okay, and mated it with the potato. So it was able to survive the certain disease or environmental factors. Another example would be the lion and tiger. It creates the liger. Okay, mating together, the apple and the grape. Then we have inbreeding. Inbreeding refers to breeding an organism with genetically similar traits to keep it together. So it's generally staying within the family. Dogs that are bred together to keep it pure or keep its trait strong, that would be inbreeding. Now, the problem with that is if you have the same genes, you increase the chance of passing along a genetic disorder that may yield blindness or physical deformities, such as problems in the joints. Variations refer to changes. These are differences between individuals of a species. These differences in genes give us different physical attributes. For example, humans have blonde hair and brown hair, black hair. This is a variation or a difference or a change. With birds, you can see a picture there. Different finches may have different size beaks. It's still a finch, still a specific species of a bird, but you have different size beaks. So this would be a bird kind. Okay, so inbreeding, remember, it's mating together in the family, decreases variation. Cloning. Cloning is the process of creating an organism that has the exact genetic copy of another thing. This could be a plant. This could be an animal. Identical twins are a natural clone. So a clone is referring to a copy, a group of cells or organism that is genetically identical, and this can be a result of asexual reproduction. They will have the same exact copy of DNA. So the reason why sexual reproduction wouldn't be an example would be because you're getting some from mommy, some from daddy. Okay, so there's going to be variation there. So asexual, if a paramecium or a bacteria, okay, one splits again, it's an exact copy. So that would be an example of a clone. Okay, so how is cloning done? We're going to go through the steps in just a moment, but to give you an overall basic view, a single cell is going to be removed from the parent organism. Generally, what will happen is scientists will take a stem cell. A stem cell is found primarily in the bone marrow. That stem cell has genetic information of the whole body. If a scientist takes a skin cell or a bone cell or a muscle cell, primarily those cells are going to have genetic information just for that type of cell. But if they take a stem cell, which has genetic information for the whole body, that increases the chance to clone an organism. So cloning is done through and by stem cells. The entire organism is grown from that cell, and that cell contains the whole DNA of the whole body of the whole organism. And each cell in the body has the same DNA, okay, but cells. Now, we talked about Dolly being the first mammal. It was not the first animal. They experimented on frogs and other animals as well to be cloned. Now, Dolly, the cloned sheep, had the same exact DNA as her mother, okay, and she did not have a father because they took the stem cell and they replicated it. Remember, this is a form of asexual reproduction, meaning there is no father and mother. There's not a male gamete or female gamete. There's only one genetic parent, and that comes from the original. Now, here are the steps of cloning. Specifically, first, the egg is removed from the female. So we have an egg removed from the female, which contains the female's genetic information, so the 23 chromosomes, and it's waiting for the male gamete, right? So what they do is they take the female egg and they remove the nucleus of the egg. So they get rid of the 23 chromosomes. So it's empty. Then that goes to step 2. They take the body cell, the stem cell of a different person, or the person they want to clone, or the animal they want to clone. They take the genetic information, which is 46 chromosomes, 23 pairs. So this is a complete human person, or animal, or plant, and they take that genetic information, they take the DNA, the nucleus, and they remove it from the body cell, the stem cell, and they put it into the female egg. So here's step 3. They put the body cell, the stem cell genetic information, the nucleus, into the empty female egg. That's step 3. Now step 4, the egg is charged with electricity to start mitosis, to start cell division. Once cell division starts happening, they take that egg and they put it into a surrogate mother, into a woman that will allow the baby to grow inside her. And then when the baby is born, you have your clone. Now, humans have not been able to be cloned. Humans have not been cloned, but this process has been. Now, the process of having a surrogate mother or woman grow another person's child, that does happen, but not in the context of cloning. Not yet, anyways. I want you to look over the benefits of cloning, and we're going to actually be talking about that. And then I want you to look at some of the problems or risks of cloning.